Okay, Gavin, when we finished the seminar on February 11th, there was some questions from the audience remaining, and we figured that we'll do a video meeting and try to answer the remaining ones.
Absolutely.
So for the first question, does the Biopixlar need to be housed in a Class 2 laminar flow hood to maintain sample sterility?
Good question. So, yeah, I mean, when working with cells, you obviously want to be in a sterile environment, as possible, especially if you want to grow the substrate and the tissue afterwards. The Biopixlar itself, because we're positioning at the the micron level, is actually housed on an optical table, which removes vibration and the kind of ability to disturb things while you're working. So rather than putting it into a laminar flow bench, we generally recommend that the entire system is housed in a cell lab facility, which would then be rated for working in a sterile kind of aseptic way.
Okay, so the next question here, can a Biopixlar work with higher density SBS plate types?
So that's another good point. What we would typically do is and as probably we've seen in the presentation, we generally work on like gridded surfaces. So things where we actually can see exactly where things are, largely so that we can actually go back and check, later on, see how things are growing, how they're developing. However, it is perfectly possible to work with these kind of well plate formats. We do have a fairly kind of easy kind of positioning way of handling the print head. However, it does require some sort of footprint areas. So the plates that we have tried and work on are like the 6 well plates, the 12 well plates. These are both kind of perfectly fine. I think beyond that it would require investigation to really see, but 6 and 12 shouldn't be a problem.
Okay. Okay, next question comes from Saeed Al-Ubaidi, and he asks if is Biopixlar suitable for any kind of cell?
Yeah, so good point. So we've worked with many different cell types. Almost all would be classified as adherent cell types. So we typically and primarily mammalian, at this point, a lot of human and some other mammalian ones, but primarily human cell types. So we have done, I don't know, I think a dozen or so. I can quickly bring up a slide that we didn't have time to get to in the presentation, which actually, so we've worked with a multitude of different cell types, each kind of being a model for either a particular organ or a particular disease type, and these are usually quite standard that most people will be familiar with. The addition to those being things like neuronal tissue digest, where we actually took biopsy samples from rats and actually with an association with a partner, they did the dissection and supplied us with isolated neurons, and then we actually printed these neurons and actually grew these neurons as well. We've also been spending quite a lot of time recently working on different types of stem cells, both iPSCs and MSCs. Again, from these ones being from a human origin, but some of these are still showing their pluripotency, some of which actually have been differentiated to show particular kind of organ-specific stem cells.
Okay. So next up is Manohar Kaduri, and they ask, does shaking of the substrate to get into incubator disturb cellular arrangement?
So when the cells are actually flown and actually attaching to the substrate, they kind of grab on fairly well. I mean, we're able to translate and move around within the kind of substrate dish without really worrying too much. We obviously have to be careful we don't crash back into cells that we've printed, because we are very close to the surface, and this goes back into this idea of having it on a vibrationally isolated system. And when we do move the sample dish, we do take care not to do anything too abruptly. And this also goes down to, adding of additional like growth media components and things like that. If it needs supplementing with FBS, for example, then this is done carefully to minimally cause distortion. But we don't notice to any degree cells being washed away or kind of structures being broken.
Okay, nice. So next question comes from Elida Nawi, and they ask, how would you think that this technology can be translated into clinical applications as the cells are printed onto a substrate?
So we need some sort of surface to grab the cells, as we kind of discussed in the webinar. We don't use any sort of binding matrix or anything to kind of house the cells in other than their native media. So it's very, as you indicate, it's very important that we have something then to attach the cells to. However, this is not necessarily meaning that it cannot be done in clinical applications. There are plenty of different supporting membranes and other things which are clinically used, both in surgeries but also in transplantation technology for helping for either bone growth or regenerating particular areas. Having a substrate doesn't necessarily rule it out of a clinical area.
Okay. Malcolm Lee asks, does the Biopixlar technology mean the end of scaffolds for 3D printing?
So I think one thing which is very true within the field of 3D bioprinting and 3D printing in general is there's no one size fits all. It's a very broad range of different technologies trying to achieve actually quite different results. Some focus more on very macroscopic structures where actually the goal being to have something like a very structural component. For example, if you're printing like bone sections or things like that, it's very important to actually have all of those structural, very large assemblies. And then there's also ones which print very, very small things and like blood vessels and things like that, where actually you want something very soft. And so I think that and different technologies such as inkjet printing, extrusion printing, the laser-based desorption printing, photo cross-linking, these all have unique sets of capabilities. And kind of I think on the second or third slide, I was kind of excuse me, showing some of the initial ideas, but one of the kind of overwhelming ideas which came out a little bit more in 2019, 2020 is why don't we try to put some of these benefits together? Because there's no one size fits all technology, can we in fact stitch together a couple of these different approaches to try to gain the beneficial features of each of the technologies in the area? I mean, we focus primarily on not using any scaffolds. We focus on cell-to-cell interactions and cell-to-cell communication. We really want to establish these very reproducible, very representative microtissues, as quickly as possible and as kind of biologically relevant as possible. This isn't the focus for everybody, and I think that together all of these different technologies can work towards actually finding the biological solution.
Yeah. Okay, Weipeng Lim asks, can this technology be applied to create a microenvironment for blood cells such as leukemia cells?
So primarily we work with adherent cells, and I think that it's not a limiting factor. It's just what we currently work with. We do, as going back to the substrate question, we do use something to grab the cells. So as long as there was something which can hold on to the cells of interest, then the microenvironment can be patented. I mean, we do have this kind of unique ability to expose not the whole tissue, but parts of of an arrangement of cells to different treatments. So you could establish different types of microenvironment for the same kind of printed area, and in that you might be able to establish, the different types of environments that you might see in other kind of blood conditions. I think the things that it will come down to is how long you have to establish these environments for, how big, and that you can actually grab onto the cells that you really want to use as the basis for your model.
Okay. So the next question comes from Muli Yamwal, and they ask, would 3D printing technology be able to reproduce working cell glands such as the pancreas to assist in insulin production?
My assumption would be yes. I mean, it's and one of the things that we showed with the kind of liver example, it being fairly quick, showed essentially the power of doing things on a small scale. I mean, we use these very simplistic models of a liver, and built these different kind of structures using hundreds of cells. I mean, very, very few cells, and actually cultured them and actually monitored their secretion over time. And actually, we not only monitored those model secretions, but other versions of the same model, either with different levels of complexity, and studied the differences of these different models over time. So I think that kind of gives credence to the idea that you can print and you can do it in a way where you can allow the cells, if they do secrete substances, to actually secrete them into the supernatant. You can collect the supernatant and do subsequent analysis, with it. So I think that if you understand the biology of the microenvironment, and it's something which you have access to with regards to the correct cell types, then I think it should be possible to establish something where you can then monitor what's being excreted from the cells.
Okay, yeah. So the final question comes from Onyekachi Anyamele, and they ask, as a life science undergraduate, will I require any engineering background to explore this area of biotech?
I think one of the things that we do here, and I think is a goal for a lot of the kind of technology areas, like we really focus on empowering researchers to do their research. We want to be able to provide solutions, technological solutions, which allow you to focus on the biological aspects and the biological question, rather than knowing exactly how the technology works. And this is kind of true with the Biopixlar as well as the Biopen and everything else we work with, is that we develop the software and the interfaces to be as kind of user-centric as possible. I mean, the Biopixlar, one of its kind of features which a lot of our kind of users like, is it's all controlled with a joypad. I mean, we use things that are very, very comfortable for anybody to use. All the functions are already pre-programmed into buttons, so that you can actually just focus on achieving what you want to achieve to address the question that you want to address. The understanding of the technology is not, let's say, a prerequisite, and with regards to the technology that we develop, we're always available to help people expand on their ideas. So if they do need to understand the engineering, we can help support that.
Okay, thank you, Gavin, for taking the time to answer all of these questions. And